Hello, everyone. I'm delighted to welcome you to today's webinar that BioPredic organized in collaboration with Koma Biotech, our distributor in Korea, and Sola, our Japanese partner for cosmetic testing. This webinar is about human skin models, unveiling mechanistic pathways of cosmetic ingredients to sustain claims on finished products. So thank you all for taking this time to join us today. I'll be your host and guide throughout this webinar. Before we begin, please note that this webinar is being recorded and a link to the recording will be shared with you in the upcoming days. If you have any questions or thoughts or would like to engage with our speakers, feel free to use the Q&A box. So our first session will be presented by Roma Lalek, a commercial solution designer at BioPredic. He will be presenting human skin samples from ethical sourcing to processing and supply. Roma is from the sales team at BioPredic. He has been working in this position for 5 years now and has a good knowledge of BioPredict products, especially skin models. So his role involves supporting clients in their research project by providing tailored solution to meet their need. The second session will be presented by Yuri Okano, a chief researcher at Sola. Her presentation is titled Cosmetic Testing, Mechanistic Insights from Ingredients to Finished Products. Yuri has an extensive experience in the Japanese cosmetic industry, where her career began with a focus on product formulations. Over the years, she transitioned into the field of safety and efficacy evaluations, where she has built a strong expertise. So her current work involves assessing cosmetics and their ingredients using a variety of methods, including monolayer cell cultures, 3D skin models, ex vivo evaluations, and human clinical trial systems. She has also been recognized for her contribution to this field, receiving awards at prestigious conferences such as the International Federation of Societies of Cosmetic Chemists. So, Roma, the screen is yours. You can share your presentation with us and start this webinar. Thank you.
Charbel. So can you hear me and can you see my screen?
Yes, perfect.
Okay, good. So thank you, everyone. So I'm delighted to be here today to introduce our activities at BioPredic International. So as Charbel said, today's topic is about our skin range of products at BioPredic. So I am Romain Laelec. So I've been working with our partners and customers using our skin products for the last 5 years, and I'll be happy to introduce that to you today. So as an overview, today I will present our solutions of skin products. So how we source it, how we work with our network that source the skin for us, our capabilities in matter of quantity of skin that we can offer to you and specifications that we can apply to our skin, our capabilities in matter of skin treatments and how we control it. And also I will make an overview of our skin primary cells and skin subcellular fractions. So to introduce BioPredic International, we are a French biotechnology company with over 30 years of experience in designing and manufacturing human and animal assay systems. So we bring solutions to the drug, cosmetic, and chemical companies in testing products with a special focus on several products. So BioPredic International has a strong global presence. So we have several production and distribution sites worldwide, so including 2 sites in Rennes and Paris in France. one site of production and distribution in Noida, which is in India, and one distribution and production site in Nashville, so in the United States. Also, some of our products are also distributed globally by several key actors of the market, such as Thermo Fisher and Lonza, and also by several national distributors worldwide. So in South Korea, our distributor is Kama Biotech, and they can supply you all the BIOPREDIC catalog in your country. So our solution, as I said, is to provide assay system for the pharmaceutical, cosmetic, and chemical industry, with a special focus on 3 main range of products. So first range of product are the liver products. So we supply human hepatocytes, the HepaRG cell line, which is famous, the HepaSH, the liver subcellular fractions, silenzome, and also cell culture medium and supplements. We also work with fluids products, so we can provide fresh and frozen whole blood and derived products such as plasma and serum, blood cells, so PBMCs, lymphocytes and monocytes, urine and intestinal fluids, and of course, the topic of the day, so skin products. So with our fresh and frozen skin range of products and skin cells and skin subcellular fractions. So first, I would like to make a zoom on the skin sourcing. So as I said, we have an extensive clinical network in France and in the United States. So this is one of our key strengths. So with this vast clinical network, we are able to collect more than 3,000 human skin samples a year, which allows us to produce more than 10,000 skin samples that are used globally by many actors across the world, such in the paper that I attached about an OECD study ring trial on caffeine using the BIOPREDIC skin product. So we worked with many key actors, such as Charles Rivers, Bayer, BASF, and Eurofins, but many others as well. So at BIOPREDIC, we prioritize the ethic and regulatory compliance. So all tissues are collected in full adherence to the current regulation in France, with informed donor consent. We also maintain a strict confidentiality and transparency of our sample. We have many permits that I will describe now. So first permit is DC permit, which is a permit to collect, store, and use human biological sample for research use only. We also have the HACI, which is accreditation of session. So this allows us to sell human biological sample for research use only. And we have the import and export permit that allows us to export the skin to South Korea. So as said, it's very important, but I said it again, each skin donation are collected from volunteers donors, volunteers that gave their full and informed consent prior to the surgery. The samples are coded, so the confidentiality of the tissue is maintained all the time, and the donors are not compensated for their donation. So we have a simple but efficient workflow with our collection site. So first, they gather the consent of the donor ahead of the surgery. Then the surgery is done by the clinics, and the biological sample is packed and sent to the BIOPREDIC laboratory. Then BIOPREDIC makes the process of the skin under the specification of the customer, and within 24 hours. Then we ship it to South Korea. So this extensive collection network allows us to collect many diverse skin samples. So first, we can collect from many body sites. So we have abdomen skin, breast skin, arm skin, thigh skin, and also scalp, eyelid, and foreskin. With the skin, we can provide many clinical information, so including medical history of the donors, if they take medications, what are their lifestyle habits, or if they have any diabetes. We have a diverse demography. So we collect from donors from 18 years old to 80 years old. We have a diverse ethnicity, so Caucasian, African, and so on. And many phototypes, so from 1 to 6. So this is our capabilities in terms of processing. So we start from the whole skin, so skin with adipose tissue. So either you need it with the adipose tissue and we send it like that, or we remove the adipose tissue. Then we can supply the skin without the adipose tissue or dermatome with a specific thickness depending on your need. Or we can also make from this dermatome skin a dry stratum corneum. So all of our skin are available in 2 formats. So either as sheets, so from 12 centimeters square to up to 200 square centimeters square, or we can make skin disc from 5 millimeter diameter up to 40 millimeter diameter. All our sample can be supplied fresh, frozen, or fixed in the solution of your choice. So there it is. So some pictures of our skin. So you have the skin disc on the left and the skin sheet on the right. These pictures are always displayed in our certificates of analysis. So if you want to pick a sample, you know how it looks before choosing it. So speaking about our certificate of analysis, each skin comes with their set of information. So we always detail the donor's condition on our COA, so its age, ethnicity, and virological status. We make a microscopic examination of stretch marks, scars, or hair. We put a sample image of the skin sample. And for dermatome skin, we make an additional quality control where we measure the thickness of the skin. And for our OECD guideline compliant product, we also perform a transepidermal water loss QC, quality control. So for us, the transepidermal water loss quality control is important. So why did we choose this control? So the transepidermal water loss quality control is a critical parameter in OECD guideline compliance product for skin integrity assessment. The TEWL value allows us to check how much water vapor goes out of the skin. And so by doing so, we can check if the stratum corneum still performs its duty properly. So this measure is directly comparable in vivo and in vitro. And to do so, we keep a sample of each of our frozen skin that we produce, and we make the 3 steps that are mentioned in the slide. So we saw the sample, we adjust the skin at the right temperature, and then we use a condensed chamber aquaflux to make the measurements. So this transepidermal water loss is recommended in the OCD guideline recommendation. And so that's why we have chosen this quality control. We have many historical data on it. And so our threshold for this value is 15, and all skin that are above more than 15 are discarded and not kept by BIOPREDIC. So in addition to our fresh skin product, we also provide a specialized culture medium that will allow you to culture the skin for up to 14 days after the surgery. So it's a good medium that can be used in long-term studies, so such as skin regeneration, toxicity testing, and metabolism research. So that medium is commercially available and can be shipped either on dry ice, so you can keep it for 12 months, or we can send it at 4 degrees along with your fresh skin sample, so you can use directly this medium when you receive the fresh sample. So this medium is animal component free and ready to use medium. We also made some smartomic analysis on it, which is RNA sequencing. So if you need some RNA sequencing data using this skin medium, we will be happy to share it with you. So our skin range of products address a large set of applications, which are described on the screen. So fresh skin allows you to assess or address almost all the applications and almost all the skin testing possible. The frozen skin is better for the transcutaneous passage, and the stratum corneum for some other application. Also at BIOPREDIC, we offer skin primary cells such as keratinocytes and fibroblasts with some specifications that are mentioned there. So post-so viability is mentioned on the COA. Both of our skin primary cells are packed in vials of 1 million cells at passage 1. And also we put pictures of each batch of cell on our certificate of analysis. And coming soon, we plan to offer sebocytes for those who are interested. Then BIOPREDIC also offers skin subcellular fractions, such as microsomes and S9 fractions, which are also which are in vials of 0.5 milliliter with at least 10 milligrams of protein per vials. So how to access our skin products in South Korea? As I said at the beginning, our distributor there is Coma Biotech. And after the presentation of our partner Sola, they will explain for our South Korean audience how to access the skin and how to order BioPredic skin with them. So thank you for your attention. I leave the voice to Charbel.
Thank you, Romain, for this insightful presentation. And we will happy to answer your question. If you have any question, you can ask them in the Q&A box below in the Zoom. Now we can switch to the second presentation of this webinar. So Yuri, the screen is yours now.
Thank you, Charbel. So my name is Yuri Okano. Good afternoon, everybody. So I'm talking from Japan, Osaka today. So I will present my presentation. I will share my slides. Can you see it?
Yes, perfect.
Okay. So I will talk about cosmetic testing method, mechanistic insight from ingredients to finished products. So in the field of evaluation, there are roughly two categories. One is in vitro study and another is in vivo clinical study. Which test do you choose in your situation? I will show you the detail of the process of the development of functional cosmetics. So these are the flow. First of all, we have to decide what kind of function the final products have, how to differentiate from other products from another companies or all ourselves products. After that, we start to find out the functional ingredients, so-called active ingredients, that fit to the concept. If we find something good, next, we develop the formulation containing the active ingredients. And finally, we have to check if the formulation works as we expected. But in this process, so we have to decide the concentration of active ingredients in the final product, finished products. And there are two points of the evaluation points, this point and this point. In general, we used in vitro testing here, and we use clinical testing here. Next, I will explain you the methodology of evaluation shown in detail. The simplest method is the examination from the aspect of chemistry and biochemistry. The second step is the method using target skin cells, cell culture system or three-dimensional reconstructed skin. But even in this case, it is still unclear if it works on real human skin, because there are no stratum corneum in single cell culture, and even though three-dimensional reconstructed skin model has only very fragile, very weak stratum corneum. So we cannot evaluate the efficacy, including the skin penetration process. Further, we can't evaluate cosmetic finished products in those two methods because it contains a preservative and surfactants. Therefore, we want to confirm the efficacy of finished products in clinical testing. However, between in vitro testing and in vivo testing, so there are a big gap. How can we decide the concentration of active ingredients in finished products? After my presentation, I will propose the method. This slide shows that the characteristics of clinical testing. The strong points are shown here. We can obtain the evidences that the sample finished product is effective on human skin certainly. We can get very attractive data for consumers and for marketings. But there are some weak points. We can't screen the ingredients. We can't carry out human testing without safety guarantee. And it requires clinical testing requires the large scale of volunteer to obtain statistical differences. So these are the contents of today's talk. First, I will explain you about the characteristics of in vitro examination evaluation. So there are a lot of cells we can buy from the market. You know very well about the keratinocyte and melanocyte that produce melanin and the dermal fibroblasts. These are very famous. But in these days, we can buy the endothelial cells, such kinds of that from the market. Recently, it was reported that the skin conditions are determined by not only one kind of cells, but also the communication of all cells in the skin. Therefore, when thinking of the condition of skin, we have to consider the crosstalk of these cells in the skin. Further, this single-cell culture system doesn't have stratum corneum. Therefore, in such systems, we cannot evaluate skin penetration. As the evaluation model with stratum corneum, we can buy three-dimensionally constructed skin models. These are the three-dimensional models, 3D models we can buy in Japan. The upper type is only epidermis. It contains keratinocyte only. The middle one contains the keratinocyte and melanocytes. We can evaluate the skin brightening effect using this model. Recently, full thickness model with epidermis, endodermis, and sometimes melanocytes on the market. These models are very convenient to screen the ingredients, but unfortunately, this barrier system is very weak and we cannot evaluate the finished products. These are the comparison of transepidermal water loss, TEWL, between 3D model and human skin. TEWL of our skin, healthy skin, is 8 to 15 gram per square meter per hour. However, the TWL of three-dimensionally constructed skin is very high. It means the barrier function of 3D model are very weak. Therefore, it is difficult to treat them with finished products containing a lot of surfactants and preservatives. This is the characteristics in vitro study. The strong point is we can screen the ingredients, a lot of ingredients, and compare each other. We can investigate the mechanisms of the effects in detail using the biological or a chemical method. and we can explain the differences between each ingredient. But there is some weak points. We can't evaluate the insoluble materials in the medium. We can't evaluate the finished products. And it is not possible to predict the concentration of active ingredients in the finished products. So challenges of in vitro evaluation is here. How to link the results to real effects in clinical trial using human subject. How to decide the concentration of active ingredients in the finished products. The next topic, I will introduce you the new evaluation method, ex vivo evaluation, using the excised human skin explained by Roman before. So in general, we can't obtain excised human skin easily in Japan and in Korea too, and cannot carry out especially industrial research using these human skins. In France, as Roman presented before, we can use excised human skin shown here in the industrial researches. That's the reason why we established SOLA in France. And actually, SOLA is in the laboratory of BIOPREDIC. So we can obtain very fresh skin materials from BIOPREDIC. The cells in the skin remains alive, and they have stratum corneum. Therefore, evaluation including percutaneous absorption are possible. We can evaluate cosmetic ingredients and finished products. Further, the skin has already been excised. Biological or biochemical and histological studies are possible as same as in vitro study. These are the skin discs we can use. These are the difference of thickness. This is very thin, and this is full thickness skin. So BIOPREDIC dermatomes the skin tissues and can prepare the uniform thickness skin discs. It is very easy to use in our laboratories. Sometimes we use the skin sheet. In this case, we use skin sheets for evaluation of beauty instruments. There are the comparison of transepidermal water loss between 3D model and excised human skin. The TEWL of excised human skin is 8 to 15 grams per square meter per hour, as Roman explained. So it is perfectly same as our skin. It means that this excised skin is superior tool with perfect barrier system, and we put the finished product on it. So here is the positioning of ex vivo study. So in vitro studies are superior to study the mechanisms, but it's not real skin. In clinical study, it is real skin, but we are hard to obtain the skin tissues after clinical testing from the volunteers. However, excised skin is real skin from human and has a strong barrier system. There are no ethical problem in vivo study, and we can carry out the evaluation as same as in vitro study using the biological or histological method. That is, ex vivo study is very useful tool to connect in vitro study and clinical study. As I mentioned before, it is difficult to decide the concentration of active ingredients in finished products. from the results of in vitro testing. Ex vivo study is standing between these points, like this. In ex vivo study, we can evaluate cosmetic ingredients and finished products. As the first step, we confirmed activity of ingredients using ex vivo study. Further, we can evaluate various concentration of active ingredients in the finished products and can determine the concentration of the active ingredients. That's then the clinical study can be done. Use the finished product containing the suitable concentration of active ingredients. Next, I will show you some of our results using the excised human skin. From the ex vivo study, we can obtain the data of gene expression, protein expression, and histological data and biological data like a clinical study. This is transepidermal water loss. There are two options about the messenger RNA expression. After extraction of total RNA from skin disks, we can carry out the quantitative PCR or BRB, bulk RNA barcoding sequencing, BRB-seq. In our laboratory, we will use smartomic analysis provided by Eurosafe. So BRB sequence is very unique system. In the case of quantitative PCR, we can check the changes of specific targeted messenger RNA. But when we don't know what function that ingredients show and want to comprehensive search for their effects, we use BRB sequencing. In this method, we can check more than 10,000 gene expression. Further, combination of special data analysis makes us to be able to analyze the functional characteristics of the skin treatment. In this slide, I will show you the results of BRB sequencing after UVB radiation. In this assay, over 10,000 genes were detected, and the expression of 240 genes were different with or without UVB radiation. The red and then gene ontology analysis were performed for DEGs and picked up the GO terms, which was higher correlated. The red and blue circles means the changes of messenger RNA expression. The size of the dots means the number of changing genes. For example, cytoplasmic translation, epidermis development occurred big changes in UVB radiation. So these results gave us not only existing information, but also new positioning to clarify the wrinkle formation induced by UVB irradiation onto the skin. Next, we will try to do the sample treatment before UVB radiation. Then, skin discs were collected and analyzed the BRB sequencing. In this study, differentiate expression gene were only 178. It is less than UVB radiation. It means the sample treatment suppressed the change of gene expression induced by UVB radiation. Interestingly, we found that the sample act the regulation of RNA splicing and the process of serine transport by geoanalysis. This information helps to clarify how the sample works on the skin discs. Of course, BRB sequencing can detect the alternative of wrinkle-related genes such as interleukin-1 alpha, interleukin-6, CXCL8, type 1 collagen, and MMP1. After UVB radiation, these inflammatory cytokine increasing, but collagen synthesis decrease and the MMP1, that is collagenase, is increased. However, the sample treatment before UVB radiation shows the very yellow column. It shows the decrease of the cytokines cytokines of inflammation and recover the collagen synthesis and decrease the collagenase expression. That means this sample should be expected as an anti-aging, anti-photo-aging ingredients. Further, I will show you the evaluation using the finished products. I will show you the example of using the finished products. We treated skin discs with cosmetics on the market. After 7 days, we collected the skin discs and carried out the immunofluorescent staining of procollagen histologically. This is the result. This is control skin discs, and this is the finished product results. You can see a blue dot. Blue dot means a cell nuclear. There are a lot of blue dots. This is epidermis, and this part is dermis. And green fluorescent means the newly synthesized procollagen. You can see here, this is placebo cream. This is cream containing vitamin C, and this is cream containing retinol. So you can see the very strong fluorescence in vitamin C cream and retinol cream. It means the 7-day treatment of vitamin C and retinol cream, that is finished products. So these finished products increase the collagen synthesis and increase the procollagen in the dermis. So finally, I will show you the histological changes after UVB radiation. These are the results of skin pigmentation induced by UVB radiation onto the skin discs. We irradiated UVB 3 times onto the skin discs. After 5-day cultivation, we can see the clear pigmentation on the skin discs as similar of our skin. So this is the results of the irradiation onto the human skin. The right figure shows these skin sections. This is without UVB radiation. This is UVB radiation. The black dots means the melanin granules. We carried out Fontana-Masson staining and visualized the melanin contents. In the case of without UVB radiation, you can see a small amount of melanins, but after UVB radiation, you can see a lot of brown dots that contains some melanin content is very high. So on the other hand, sorry, you can in that case, you can evaluate the melanin contents from the skin discs. If you put your sample in this screening system, you can predict the ability of your ingredients, if it shows the skin lightening efficacy or not. And these are the results of protein expression in skin tissues induced by UVB irradiation. The upper side is sham UVB irradiation, that means without UVB irradiation. The lower part is with UVB irradiation. This is interleukin-1 alpha, this is MMP1. You can see the increasing of interleukin-1 alpha and the MMP1 expression, protein expression by UVB radiation. And this is procollagen. In the case of without UVB radiation, you can see the procollagen synthesis here, but after UVB radiation, the signal of procollagen decreased. That is the first step of the photoaging. This is the characteristics of the ex vivo study. The strong point is all cells are alive in the skin discs. We can treat with both ingredients and finished products. The skin responds like the real skin against the stimuli, like UVB radiation or sample treatment. The function, including penetration, can be evaluated using the skin discs. The inside changes of the skin can be observed by biological method or histological method, but it has some weak points. They can't be cultured only, they can be cultured only 10 to 14 days. We have to finish our examination in this period. The freshness is very important. So finally, I will summarize my presentation. The summary is here. The data of clinical study is the most valuable, but only the data from outside of the skin can be obtained. In case of in vitro study, it can investigate the mechanisms of the efficacy very details, but only it can evaluate the effects, including proteinous absorption. Ex vivo study can observe the response inside the skin, including a percutaneous absorption of test samples. Ex vivo study can determine the concentration to be used in the finished formulation. So this is my last slide. I would like to propose this evaluation flow for the development of functional cosmetics. First, please check the messenger RNA expression of your ingredients. And next, please check the protein expression using your ingredients. After that, please use the ex vivo study and determine the concentration to be put in the finished formulation. And after that, lastly, please confirm the efficacy using clinical trial. If it shows superior efficacy in vivo study, probably it should show good results in clinical study. And after that, please launching your products. That's all. Thank you for your attention.
Thank you, Yuri, for this excellent and informative presentation. If you have any questions, please don't hesitate to use the Q&A box below in the Zoom account.